Hi everyone, can you hear me okay? Okay, I left most of my voice at SFN, so I hope you'll bear with me. First, I want to start by thanking the organizers for including me in this fantastic lineup. I'm honored to be here. And I'm excited to tell you about some of our past and recent work. Some stimuli carry innate emotional valence. No training or learning is required to have emotional responses to them. Other stimuli are initially neutral unless they're paired with an emotionally significant stimulus. For example, a picture of the Twin Towers might have been initially neutral, except for us associating it with the horrific events of 9/11. Now, when shown this image, it may evoke emotional responses. There's been a lot of controversy about whether emotion is experienced in animals in a similar way or in the same way that it is experienced by humans. Of course, there's no way to ever know how an animal experiences emotion. There's no way to ever say whether it is the same as a human's experience. There's also no way for us to truly know whether another human's subjective experience of their emotional state is the same as another's. So even though I'm interested in emotional processing, what I actually study is motivated behaviors, because that's what we can measure. So the question that I'm really interested in is, how do we assign motivational significance to sensory stimuli? For example, if there was a gunshot, a bang, if you were a war veteran, this might evoke a negative affective state. But if you're Usain Bolt, maybe that makes you think of all the gold medals you won and evokes feelings of excitement and reward. But if you're presented with a stimulus over and over and over again with no consequence, like someone who works in an arcade, perhaps you habituate and then there's no response at all, and you have you're basically bored in the presence of the stimulus. And so this kind of goes back to the two-dimensional theory of emotion, initially proposed by Lang, where on the y-axis we have intensity or arousal, and on the x-axis we have valence or hedonic value. So here, when you have low intensity, this is neutral or bored. So if we think about this framework and also lay this alongside a different framework, the two-factor theory of emotion, which discusses how when a stimulus is presented, the first thing that happens is you determine if this is important. You're constantly bombarded with a barrage of sensory information, and you have to determine what is important, what should you pay attention to, what should you filter out. And so the first question being, is this important? Is it salient or arousing? And you might think of this as the absolute value. If it's not, then it's neutral. If it is salient or arousing and important, the next question is it good or bad? Is it positive or negative? Is it something that we should avoid or something that we should approach? And so this kind of goes back to the Schachter and Singer two-factor theory of emotion. And so today I'll be focusing on this part of the question. Where and how do we determine if something is positive or negative? So the amygdala has been long appreciated as a region that's important for emotional processing. This has been known from days of Brown and Schaefer over a century ago, then termed psychic blindness by Kluver and Busey when primates that have had amygdala lesions or temporal lobectomies show a blunted affect, no response to snakes, for example, and Larry Weisskranz then sort of narrowed it down to the amygdala. In humans, the famous case study patient SM, when she had bilateral lesions of the amygdala, she lost fear responses to being mugged, being able to recognize emotion in other people's faces, but not the production of autonomic arousal. If you suffocate, if you put too much carbon dioxide, she would have an autonomic response that suggests that the autonomic response, the arousal, and the emotional response are separate. So in my lab, we study mice, and if you take a cross-section here, this is the basolateral and central amygdala, and if we blow this up, this is what I'll be focusing on today. And so the outline of the talk will be, where do circuits that encode positive and negative valence initially diverge in terms of the stream of information coming through. Of course, there are many different regions throughout the brain that encode positive and negative valence, but how is this initially parsed? How do positive and negative circuits locally interact at the level of the basolateral amygdala? And then I probably won't have time to get into it, but we're also interested in how circuits orchestrate competing motivational signals. So if we think about the basolateral amygdala and the central amygdala in a very simplistic way, since this symposium today is covering many different brain regions, I'll just give you a very, very broad overview. Some have likened the basolateral amygdala to a cortex-like region. It doesn't have the laminar structure that a cortex does, but it is 90% glutamatergic neurons and is a site of associative plasticity. The central amygdala, in contrast, is striatal-like in that it is also largely composed of GABAergic medium spiny neurons. And so many have conceptualized the amygdala as a miniature primitive brain within the brain, a little cortical striatal loop that's more well conserved throughout evolution. So if we think about how information is coming through from sensory systems, as we heard about this morning, and into more limbic structures for processing, there's a lot of evidence that supports that the basolateral amygdala is a very, very well-positioned site for divergence of circuits that process negative and positive valence. We know these circuits must diverge since they produce such different behavioral outputs. And so where does this happen? We know that neurons in the basolateral amygdala encode positive and negative valence, as well as salience in general. We know that sensory information converges of all modalities into the basolateral amygdala, and it's a relatively early site in the stream of processing for this to happen. We also know that learning induces plasticity. Associative learning induces plasticity in the basolateral amygdala, and specifically the idea is that Hebbian plasticity occurs in inputs carrying information about the conditioned stimulus, often in our field, this is an auditory tone, and then when we pair this with either a foot shock, an aversive unconditioned stimulus, or sucrose, a positive unconditioned stimulus, that will get plasticity. And so following up on the beautiful talks that we've just heard from Carla Schatz and Graham Collinridge, I don't need to tell you anything more about long-term plasticity, except for that one measure, one proxy for plasticity, though it's certainly not perfect and certainly not comprehensive, is the AMPA to NMDA ratio. What it is robust and replicable, and one that has been reproduced many times in our field. So generally speaking, as you've already heard, following long-term potentiation, you'll see an increase in AMP receptor composition, function, currents, number of AMP receptors, and following LTD, you'll see a reduction in AMP receptor-mediated currents, and this will be seen as an increase or decrease in the AMPA to NMDA ratio, assuming that NMDA receptors are being held stable. And so multiple groups in our field have shown that following fear conditioning, you can see that there is an increase in AMPA to NMDA ratio when we stimulate putative thalamo-amygdala synapses coming into the basolateral amygdala following pairing of a tone and a foot shock. I should say Simon Rumpel and Roger Clem and Robert Malino and Joe Ledoux's lab and Rick Gairdner's lab respectively have shown that this is mediated by postsynaptic changes in AMPA receptor composition and function in calcium permeable AMPA receptors. As a graduate student, I showed the same thing in reward learning, in that following pairing of a cue and sucrose, that you'll also see an increase in AMPA-NMDA ratio is also mediated by postsynaptic increases in AMPA receptor-mediated function. Okay, so if I leave it at this point, it raises a very important question. How can the same mechanism underlie fear and reward conditioning? I mean, the basolateral amygdala is involved in many, many different functions, and so maybe it's true, maybe we're seeing this because it's not actually important in valence processing, and it's just determining whether something is salient or arousing, if it's important or not. And that's why we see the same the similar mechanism for changes following fear-reward conditioning. But it's also possible that the basolateral amygdala projects to many different regions throughout the brain with distinct functionality. And maybe the amygdala is the site of valence assignment, a source, the site at which there's a fork in the road, so to speak, of information processing. And there's a lot of evidence to suggest this as well. And while this is a simplified summary of what we know, I'm happy to discuss the complexities that are arising. And some of our new unpublished data that I'll touch upon at the end of the talk can speak to this as well, why there might be some clarification on the recent controversies. But to keep it simple, for now, I'll just say that what we do know is that the central medial nucleus of the amygdala is important for the expression of fear. And optogenetically or chemically stimulating central medial neurons evokes freezing responses. And that if you disconnect the basolateral amygdala projection to the central medial nucleus, you'll also abolish fear expression. Of course, as I was mentioning before, there are other roles of the central amygdala, including Q-potentiated feeding. On the other side, what would be a reasonable target for driving approach? The nucleus accumbens is a likely target. There are many, many, many more studies, but some of the first come from Barry Everett's group, showing that the nucleus accumbens is important for reward-related behaviors. More recently, we've learned that optogenetic stimulation of amygdala projections to the nucleus accumbens supports self-stimulation and also place preference. And so at this point, we wanted to determine whether this was the case. Is this the fork in the road? Is this the site of divergence for circuits processing positive and negative valence? So I'm extraordinarily grateful to have had the opportunity to work with a very talented graduate student, Preneeth Danbury, and a wonderful postdoc, Anna Baylor, who's now got her own faculty position in Bordeaux. And they joined my lab before I had any furniture or equipment. And we hypothesized that the basolateral amygdala projection target that when we pair a conditioned stimulus with a negative US, that you'll see synaptic strengthening onto neurons that project the central medial nucleus of the amygdala. And so for the rest of the talk, I might refer to these neurons as central amygdala projectors or CEM projectors. They are neurons where the soma is in the BLA, so don't get confused. And the contrast is we also hypothesize that following reward conditioning, when we pair a cue with sucrose, that we would see relative synaptic strengthening onto accumbens projecting BLA neurons. And so what we did initially, just a very simple experiment, was to label these different projections with retrobeats, retrogradely traveling beats to target which neurons were going where, trained animals under either a fear conditioning or reward conditioning paradigm, and then we stimulated electrically and patched these cells. Pretty simple. What we found was interesting, and there were some things we expected and some things we didn't, so I'll try to highlight the thought process that we had. And so at this point, this is old news, but what we found was that following fear conditioning, synapses onto CEM projecting BLA neurons underwent the potentiation, an increase in amp-attendity ratio. Interestingly, following reward conditioning, we saw a decrease in amp-attendity ratio, so a double dissociation. Importantly, I want to point out that in the reward conditioning part of this task, we are food depriving the animals prior to the task, which we didn't really think too hard about. It's just what we need to do to make the animals learn the task. But this comes up, this is an important foreshadowing clue for later. Okay, the opposite happened in BLA neurons that projected the accumbens. Following fear conditioning, we saw a decrease in amp-attendity ratio, and following reward conditioning onto accumbens projectors, we saw a potentiation. But again, one thing I want to point out is if you just look at these naive groups, the naive food-restricted animals, the naive groups, the only difference between these groups is that this group is food-restricted, and you can see that there is potentially a difference in the amp-attendity ratio when we food restricted. This is not what we set out to look for, but I'll come back to that later. Okay, so what I've shown you so far is that there's a double dissociation of learning-induced synaptic strength onto these different projection-defined populations of neurons. But, animals learn things. There's all sorts of different changes throughout the brain. Is there a causal relationship? Is this the hub of, is the basal lateral amygdala the hub of translating sensory information into a motivational signal? If it was, then we would expect that in naive animals, separate from any plasticity, if we just stimulated these projections alone, we would see motivated behaviors of different valence, approach or avoidance. And so, in collaboration with Ian Wickersham, we expressed rabies virus into either the accumbens projecting or central amygdala projecting populations of neurons and tested animals to see if they would perform intracranial self-stimulation. Consistent with previous studies, what we would have expected to see and what we did see was that stimulating accumbens projecting Belay neurons produced self-stimulation, approach, and positive reinforcement, but if we photostimulated central medial projectors, what we instead saw was a real-time place avoidance, consistent with promoting aversion. So, fortunately, I'm following Graham Collingridge, and this is a very, very, very simplified version, but thinking about the most simple type of garden variety, Hebbian, NMDA receptor-dependent LTP, if this is mediated by an NMDA receptor-dependent mechanism, then what we would expect, what we need to happen is glutamate needs to be released, we need depolarization to happen, we need this magnesium blockade to come out of the NMDA receptor pore so that calcium come in and do its thing. So if this was an NMDA receptor-dependent mechanism, then we would expect that hyperpolarizing postsynaptic neurons would prevent learning during this specific time window. This does not prove that this is the right mechanism, but is consistent with this model. So what we did next was we used a dual virus approach, a retrograde viral vector expressing CRE, and which we got from Eric Kramer, and injected this into the downstream targets bilaterally, and then injected a viral vector that is CRE-dependent into the basolateral amygdala. This is a lot of words to say that we expressed halorhodopsin selectively into either the BLA NAC or BLA-CEM projections. And then we ran our animals on either fear or reward conditioning and photoinhibited only during the presentation of the US. So in this case, the cue is presented, co-terminates with foot shock, and we photoinhibit only during foot shock presentation. And with the reward task, we photoinhibit only when the animal is licking sucrose. And so what we found was that following photoinhibition of the central BLA to central medial amygdala pathway, we saw an impairment in fear learning. As you see here, a reduced freezing when we inhibit the BLA to CEM pathway. Then, what was a little bit surprising was that following reward conditioning, so if I was to, draw out what I expected to see, I would have expected to see the red line here and the green line down here. That was what I expected to see. That is not what we found. Instead, what we found was that we didn't see an effect of photoinhibiting accumbens projectors. Instead, what we found was an enhancement of reward learning upon inhibiting BLA to CEM projectors. So that was a bit of a head scratcher, and we kind of brushed it under the rug, like maybe this is just a technical thing, we're not getting all the, accumbens is very big, maybe it's just an incomplete population. Those things might still be true. However, we've dug into this, and this is really the launch point for several other projects. Okay, so what I've told you so far is that we've got opposite synaptic changes that map onto projection. Following fear conditioning, we see synaptic strengthening onto CEM projectors, after reward conditioning onto NAC projectors, that if we get rid of all of the learning component in naive animals and just look at what happens when we photostimulate, we get avoidance when we stimulate CEM projectors, approach when we stimulate accumbens projectors. Of course, these projection populations are not homogeneous, and I'll talk about that in a moment. But then, curiously, when we inhibited central medial projectors during fear conditioning, we impaired fear learning, but during reward conditioning, we enhanced reward learning. That was tricky. However, we got a valence-specific effect, and so, okay, that's somehow working. But is it really that simple? How does this align with everything else? And so, as an American, in 2015, I wrote, what we're thinking about when we stimulate a different population of neurons is that we're getting what this population of neurons can do, and we're getting the majority vote. This is not the physiological activity of the neurons. This is this is certainly different. And so even though we are excited about the power of optogenetic tools, we might still be observing a majority vote, and this does not mean that we are capturing everything that this population of neurons does. However, after the 2016 election, I wish I could rewrite this paragraph and include an additional clause about the possibility that some neurons are more capable of influencing other neurons, that they have more power, and that not all votes are equal, perhaps like an electoral college. And so some of our data speak to that. And more simply put, more eloquently put, optogenetic tools tell us what neurons can do, not what neurons do. Okay, so what have I told you so far? I've told you that these different projection-defined populations of neurons diverge at the level of the basolateral amygdala, but is it really that simple? And the answer is how, the question is how heterogeneous are these populations? And so in terms of defining valence encoding is a term that recently has been used more and more, and I just, everybody has their own definition. I'll tell you mine. So for me, to call a neuron valence encoding, it must meet the following criteria. Number one, it must be task responsive, obviously. Two, it must have differential responding. And sorry, I should have oriented you. Here in green on the top row, I'm showing you different possible responses to a reward predictive conditioned stimulus. On this axis, I'm showing you different responses to an aversive CS. And so if a neuron responds similarly to a positive or negative CS, then we cannot say it is valence encoding. It could be just encoding salience. And so, and finally, it needs to be independent of stimulus features. If there are somatosensory cues versus auditory or visual cues, those modalities could be mapped onto the amygdala as well. So if a neuron is specifically responding to one thing, it could be sensory modality, not valence encoding. And so those are two important things that I think really rely on in vivo recordings. So to do this, what we looked at was a very simple task. I'm going to skip this movie, but basically this mouse licks for either a cue that predicts sucrose or a cue that predicts quinine. And what Annabelle in my lab did was record from over 1,600 neurons. And this is just a heat map showing you all the raw data. I know, what are you supposed to make of this plot? Well, for me, I like to look at the raw data. Black indicates an inhibition, yellow indicates an excitation, and what you can appreciate that is consistent with previous publications from Dan Salzman's lab, as well as Patricia Janik's lab, is that there is a lot of different types of responses. There are some neurons that respond to sucrose predictive cues, some neurons that respond to quinine predictive cues, a lot of neurons that do nothing, a lot of neurons that do both. And so that's why I really want to emphasize this point about defining valence coding. But of course, Anna didn't do this didn't record from all these neurons just to replicate other people's work, although that's always nice. What we wanted to do was know what the different projection-defined populations of neurons were doing. So we used an approach called phototagging, and this was developed originally by Susanna Lima and Tony Zader's lab. And essentially, we have an electrode. Here we are using a silicon probe with an optical fiber. And during the task, we record spikes. Neuron A fires, great, we got it. Neuron B fires, great, we got that too. We'll use the same approaches I just told you about to selectively express channel rhodopsin into certain populations of neurons of interest. And then after the behavioral session, we'll do a phototagging session, which just means we shine light. This allows us to know which neurons are photo-identified. Very importantly, there's a really, really essential caveat to consider, which is recurrent excitation. This is a huge problem in cortex, hippocampus, amygdala, and so how do we know if the neuron that we're recording from is really different from, I guess it's straightforward if the neuron doesn't respond to light, that's easy. It's a non-expressing neighbor that doesn't receive input, but there are also going to be non-expressing neighbors that are not the ones we're trying to target that might receive recurrent excitation from these CHR2 expressing neurons. And so I won't belabor all the details, but I'll just say in every experiment that we do in my lab, we'll record from neurons, determine the photoresponse latency, and look at the distributions of nearest neighbors and CHR2 expressing neurons, and determine whether phototagging is even feasible in this particular circuit, and only if it is, proceed. So what we found, again, here's some raw data. What are you supposed to make of this? I guess the takeaway is here, this is BLA to accumbens projectors. These are BLA to central medial projectors, and these are BLA to ventral hippocampal projectors. And the takeaway is that while there are some qualitative differences, there is a lot of heterogeneity. But the bottom line is that basolateral amygdala projections to the accumbens predominantly encode positive valence. Central amygdala projectors predominantly encode negative valence, and ventral hippocampal projectors do not have an apparent significant bias for either conditioned stimulus. And so we've tackled some recurrent excitation controls. If we use a category-independent strategy looking at each neuron and the relative z-score difference between the response to sucrose or quinine, you can see that there's a significant difference between accumbens projectors and CEM projectors. If we plot this in reduced dimensionality space, again in an unbiased way, you can see that compared to BLA neurons overall, each of these populations has a different vector space. And finally, if we do categorize neurons based on excitation and inhibition in the conceptual framework that I initially defined, whether it's excited or inhibited to sucrose or quinine predictive cues, we also see this bias. So again, there's a significantly different bias for accumbens projectors and central amygdala projectors, but there is substantial heterogeneity. So it isn't that simple, and I'll talk about that as well. Okay, so what I've shown you so far is that BLA is a site of divergence for positive and negative valence. And we've shown this in 3 different ways. One, following fear reward conditioning, these projections undergo opposite synaptic changes. 2, optical stimulation produces approach or avoidance. And 3, projection-defined populations of neurons preferentially encode positive or negative valence in a significantly different way. But how do these positive and negative circuits locally interact at the level of the basolateral amygdala? What is going on? Why would all these streams of information meet? And so that's going to be the next part of the talk. So the first clue that we picked up that there was an interaction between these different populations of neurons came from this phototagging experiment. It wasn't what we set out to discover, but what we found, and I'm just going to use the central medial projector as an example. First, we've got our CTR2 expressing population, very, very short latency phototagged responses. Okay, this is the population z-score. Then we've got another slower latency photoexcited population of neurons. And then we have another population of neurons that is photoinhibited. In this case, the central medial projector population, you can see this is a substantial population of neurons that this is influencing, 113 units relative to the neurons that are expressing channel rhodopsin, which are relatively less. And so this whole network, this local network in the BLA is being influenced by this population of neurons. So if we look at each projection, normalize for the expression level of channel rhodopsin, and then just simply look at what neighboring non-phototagged neurons are excited by the projectors, that's in black, or inhibited by the projectors, that's in gray. This is what we see for ventral hippocampal projectors, this is what we see for central amygdala projectors, and this is what we see for accumbens projectors. So normalized for expression, you can see that central amygdala projectors have a significantly greater influence on the other populations of neurons around them. And of course, there are many other projector populations that we haven't explored yet. But comparing these two, maybe this explains, this is certainly consistent with our previous finding that the photoinhibition of BLA accumbens neurons had less of an effect on behavior than BLA to central amygdala projectors. And maybe it's because of this very, very robust recruitment of neighboring neurons. Okay, so there's been some controversy over the anatomical arrangement of different populations of neurons in the basolateral amygdala. Susumu Tanagawa has investigated this in genetically labeled populations of neurons. We are looking at this in projection-defined populations of BLA neurons. And when we label each of these populations, central medial, ventral hippocampal, and accumbens projecting BLA neurons, you can see here that certainly there are gradients, but they are still quite intermingled. So blue is ventral hippocampal projecting, central medial's red, and I so apologize if you were colorblind. And then in green are the accumbens projectors. And you can see that there are some hotspots, but that they are intermingled. Okay, so since they are relatively intermingled, if you popped your electrode down, you wouldn't know what cell type you're recording from based on the location. What would be the purpose of this? So there must be some reason. Maybe form gives rise to function. And so what are the possible types of connectivity that we could have? One possibility is that there could be mutual inhibition. This is certainly attractive. An attractive notion. You would want to select between approach or avoidance. Why would you want to do both of those things at the same time? So that made intuitive sense to me. I would have put my money on that before doing the experiments that we've done. And then just because it's another logical possibility, we included the idea of a unidirectional inhibition, an asymmetrical relationship. And then it's also possible that there's a neutral or excitatory relationship. Since the amygdala is known to be epileptogenic, there's a lot of recurrent excitation. That's also possible. So what did we find? And why should we care? So I'll come to that in a second. So the next body of work has been done by Gwendolyn Calhoun and Amy Sutton. And just for simplicity's sake, I'm just going to remind you, accumbens projecting Belay neurons, although it's not that simple, just as a reminder so you don't forget which is which. These were largely driving approach or repetitive behaviors, and central medial projectors drove avoidance. I will now also abbreviate these different populations of neurons with just an N and a C. So, okay. The next slide is a little busy. So what we wanted to do was stimulate from one population of neurons, which we did by expressing channel opsin in that population, and then record from the other population, which we labeled with retrogradely traveling beads. So here on this, the rows are which population we're stimulating, the columns are which populations that we're recording. When we stimulate central amygdala projecting Belay neurons and record from NAC projectors, what we get is a suppression. That makes sense, intuitively. That's what I would expect. If there's a threat, you should cease to perform any additional reward-seeking behavior and escape the predator right now. That's adaptive. It makes sense to me that it would work that way. When we looked within populations, okay, central amygdala projectors facilitate themselves. Accumbens projectors may be a trend, but not significant. And then what was very surprising to me was that when we stimulate accumbens projectors and record from central amygdala projectors, that we got a facilitation. What? This made no sense to me. I saw these data. I said, okay, let's do this different way. Let's try the different parameters. Let's start the stimulation train in the middle. Let's try different steps. We did many, many different types of parameters, and all I can say is that this is a very robust result that has been replicated across multiple experimenters and multiple rigs and all the parameters we can think of. And this is now changing the way I'm thinking about the system. So importantly, I do want to say that when we start this spike train, or when we photostimulate accumbens projectors before the current step, on their own, that does not cause spiking in the central amygdala projectors. That's important because otherwise we would just have a bunch of seizures. But when there's other input, when there's other further depolarization, we see a facilitation of the spiking that would be there, presumably when there's input. So to summarize that busy slide, I'm showing you this asymmetric relationship between these populations of neurons. Again, how does this fit with everything else we know? We have this double dissociation with synaptic strength. Shouldn't it be that simple? Why isn't it this pretty symmetry that we found before? One of the keys is that these animals are food restricted. So again, we saw this asymmetry here in our photoinhibition experiments, and also when we did this food restriction manipulation. There's a change in the state of what's going on. So this actually fits really well with our data. Not our initial model, but yes, with our data. Okay, so why might the brain work this way? Why would we want to have asymmetric unidirectional relationships like this, or biased relationships like this? If an animal is engaging in reward-related behavior, be it social interaction, feeding, drinking, this invariably exposes animals to greater threats. Particularly in mice, these prey animals, if they go out to forage or hunt for crickets, they are leaving the safety of their burrow. They should be primed for escape. So perhaps, since reward seeking is in nature generally inherently risky, priming escape could be a good insurance policy, and that is why we've evolved to be this way, perhaps. But what about when homeostatic needs change, conditions change. For example, a lioness would rarely attack an elephant in broad daylight without a lot of support. A baboon would rarely forage in water for risk of being attacked by caiman. But in resource-scarce conditions, these animals will behave differently. So we wanted to do something, look into this. We food-deprived animals for 24 hours, and what we found was sort of mind-blowing to me, at least. What we previously saw as a robust suppression, when we photostimulate central amygdala projectors and record from accumbens projectors, we now see as a robust facilitation. So qualitatively changed the direction of this interaction between these populations of neurons. And then I'll just summarize. Here we also see a subtle change, a facilitation, which was not significant before. And then this part stays the same, facilitation, facilitation. And so this was very exciting to us, but this is all on a slice, how do we know this really pertains to the living animal? And so, along with Zhang Chang, a graduate student in my lab, we have now performed deep brain 2-photon imaging in the basolateral amygdala and projection-defined populations of neurons, tracking individual neurons across days and across conditions. So we've implanted a grin lens, expressed GCAM6M only in accumbens projectors using this dual virus approach that I just told you about. And then what we see is that if you look at these representative neurons here I'm just showing you a couple of representative fields of view. The yield that we're getting is relatively sparse. That actually helps us to track individual neurons with more confidence across days. And what you can see is that in the sated condition, the animals are well fed, and we put them on, and we just look at the number of transients throughout the session. And in this session, we're not really training them, we're not asking much of them, we're just asking them to hang out, occasionally lick some Ensure. But the responses that we're seeing aren't necessarily time locked, but prior to food restriction, you can see subdued activity. Upon 24 hours of food deprivation, you see a robust increase, and when we resate them, give them a couple of days back on food, you can see that this goes back to baseline. And so here I'm just showing you some representative examples, and next I'll show you when we track the individual cells across days. Here, what we can do, we perform motion correction and then statistics to ensure that we are indeed tracking the same cells across days, which again is easier, as you can see with our sparse expression levels. And we can see, okay, across the session of the cells that we've recorded so far, what is the number of transients that we see, and follow these individual cells across days. And you can see that while there is some heterogeneity, which is to be expected from what we've seen with our physiology, you can see a robust increase in transient frequency. So we would interpret this to be general firing activity and bursting. When we put them back on food, it goes back to baseline. And this is consistent with our ex vivo electrophysiology data, showing that following food deprivation, we see an increase in firing rate. Okay. Excitingly, in the converse, following food deprivation, we also see a decrease in the transient frequency in the central medial projector. So this is, again, the opposite population, the population that we've linked to aversion and defensive behaviors. And what we see here is that these neurons are relatively active prior to food restriction. Following food restriction, these neurons go almost silent, some of them dropping basically down to floor. And so that is very exciting for us. And what does this all mean? So I'm nearly out of time. I would love to get some questions, so I'm going to begin summarizing. So basically what I've shown you is that there's this relationship between these competing BLA to NAC and BLA to CM circuits that is state dependent. Following just 24 hours of food deprivation, which is something that prior to this experiment we did without really thinking about. It's just what we did to get animals to behave and learn the task in one day and drive their motivated behavior that we're trying to get. But what we're really doing is changing the intrinsic local circuitry in the basolateral amygdala. And I've also shown you with 2-photon imaging, chronic 2-photon imaging, that this is also seen in the in vivo case. So, all right, if we take this back to our initial model as conceptualized by Lang, this is a symmetrical relationship. The intensity and arousal of positive and negative cues is symmetrical. But what I've shown you here is evidence that this, at least in the basolateral amygdala, is asymmetrical, and maybe that actually fits more intuitively. You know, if you were engaging in some reward-seeking behavior and there's an immediate threat, you have to drop what you're doing. You have to run and live to eat or mate or drink another day and escape the predator and survive. And so that makes sense. But what about in the food-deprived case when your homeostatic needs are changing what the greatest threats on your survival could be? So perhaps when you are in a state of homeostatic need, we are shifting the fulcrum of this curve, and part of the circuits that I think might be involved in that are those that lie in the amygdala. And so I think I should just end there, and I'll leave this question for another day. And I'll just say that although the brain, everything's interconnected throughout the brain, and my lab and many other labs, of course, interested in studying valence processing throughout the macrocircuitry of the cortical limbic system, amygdala circuits are very important. From a basic science perspective, they are the simple circuits that are the fundamental building blocks for understanding more complex circuits. And from a translational perspective, the phenomena that we might find to be true, the mechanisms that we identify in the basolateral amygdala, because they are so well conserved across evolution from even reptiles all the way to humans, certainly from mice to humans, it increases the likelihood that things that we find in the amygdala in our basic research can be relevant for improving the condition for mental health and other conditions of humans in the future. And so with that, I'll thank the rest of the people in my lab, my collaborators. I tried to mention people along the way, and people who've given us reagents and our funding sources, and you for all of your attention. Thanks.